It's my pleasure to introduce Dr. Ahmed Elmokadem. He's a senior scientist at Metrum Research Group. Dr. Elmokadem's work focuses on physiologically based pharmacokinetic modeling and systems biology. He'll be speaking to us on PBPK virtual population generation using open source methods. Please welcome Dr. Elmokadem.
Hello, everyone. Yeah, thanks for the introduction. Earlier, I asked Ankit to introduce me as His Royal Highness, the Lord of Westeros, but for some reason, he didn't go through with it, so I'm upset, man. I'm sad. All right, so yeah, thank you all for sticking around this late in the day. You guys are heroes. I'll try my best to make it worth your time. So I'm going to talk about the PBPK virtual population generation using open source methods. And before I start, I'm going to address the elephant in the room, which is what the H I mean by virtual populations. Because I know that different people think of the virtual populations in different ways, right? So what I mean by virtual population in this context is within the context of PBPK modeling or physiologically based pharmacokinetic models, which is a bottoms-up approach. So a population that captures the physiological variability, right? So if you're from a QSP perspective and have a different idea about virtual populations, please don't yell at me. All right, so why virtual population? This might be intuitive to a lot of people here, but I'm still gonna explain it. Why do I care about a population? If we handle a population as a monolith, like here, everyone here has the same purple color, what you're gonna get is just like a mean prediction, right? Which could be nice. So this tells us that if the Cmax is what we're looking for, it's reaching the therapeutic index and it's there for a little bit, so that's good, right? However, this is not reflective of reality, right? So in reality, people are different and people interact differently with the drug. So if we want to capture reality, we will better capture this sort of variability. Because if you see here, you can find that this one individual, for example, could go beyond the therapeutic index, leading to safety issues, could impact also efficacy and other things as well, right? So this is why we care about populations. This is why we are striving to capture a population. Okay, so the PBPK model structure. So what does a virtual population mean in the PBPK sense? Looking at a typical structure of PBPK models, this is, if you open literature, a paper, PBPK models, you're gonna see something similar to this diagram here, right? So you have all of these compartments, and the difference between PBPK models and empirical compartmental models is that these compartments actually reflect actual physiology, right? So each of these compartments is an actual organ in the body, right? So, and yeah, again, before you start yelling at me, yes, there should be arrows coming from the arterial blood into the pancreas, stomach, right? So my bad. Okay, and another thing, so these organs, they are actual organs, they have volumes. So volumes is something that you want to capture to be able to monitor the concentration of drug as it passes through these organs. And also, like all of these lines here are monitored or regulated by the blood flows, right? So this is another physiological thing that we need to build this. So what do we need to build this thing? So system-related parameters, which are the volumes and blood flows I talked about. Other things like enzyme expression, for example, right? So these are system-related. They are related to the human or whatever system or physiological system we're looking at, right? Other things that we need are also drug-related parameters like lipophilicity of the drug or pKa or partition coefficients, how it partitions between the blood and the organs or the tissues, right? I would also add like a third category maybe. It's kind of a gray area, but these are the components that are impacted by both, right? Like for example, clearance. Clearance is impacted by the drug properties and the physiological properties, right? So when I talk about virtual population in a physiological sense, I'm talking about how can we generate the population or the variability based on these system-related parameters, right? You don't expect lipophilicity of the drug or pKa to change if you change the physiological system, right? So these are gonna be the same across individuals. Okay, so how do we create that? How can we create an individual? Well, thankfully, we have a lot of data about human physiology. We've been studying it for a while now, right? So we know a lot about it. And one of the things that we know about the physiological system is that, for example, if you look at organs, we have a pretty good idea the distribution of weights, for example, or volumes of these organs among humans, right? So we can have things like this. And now, how do we make up an individual out of this information that we have? You might be tempted to say, let's just do random sampling, right? I know you're thinking it. So this is the simpler, most direct approach, right? Let's just do random sampling from all of these and put this individual together and just run simulations with it, okay? So what are the issues here, right? Can someone figure the issue? This is turning into a lecture. So if you look at this individual here, you can sample an individual that is not realistic, like someone with a big kidney here, regular lungs, small liver, right? This is not a realistic individual, okay? So this independent sampling is not gonna do it, okay? So we don't want to do that. So the alternative is do conditional sampling. Conditioned on what? On something. This something I'm going to talk about in a minute, but this something is a safeguard that makes these parameters that we're generating correlated. They need to be correlated somehow, right? So there's conditioning keeps that correlation. So now we have a more representative individual, okay? So that's what we want. Okay, so how do we create this? What do we condition this on? There are different ways. So one way is published in this paper by Willman et al. In 2007. And basically what they're doing here is that they developed an algorithm that would generate these physiological parameters or these volumes and cardiac output. So cardiac output is used to calculate the blood flows as fractions of the cardiac output, okay? So what they used is that they scaled this based on height, okay? So this might be a little small. I don't know if you can see this clearly, but let's just look at this box here, the inner box here that create the individual one. Basically what this is saying is that we're gonna condition our sampling according to the age, gender, like something that we condition this on, like age, sex, race, right? So we're gonna pick a specific age, sex, and race, and then the algorithm, what the algorithm does is that it looks through the databases that we have, right? And looks at the database, like for example, NHANES database. It's a huge database that has a lot of anthropometric information about humans, right? So it looks at a specific slice, a specific age group with a specific sex, and race if you have race information, and then looks at the boundaries of the heights and weights of these individuals, right? If the picked up height or weight is beyond this database, then it's gonna error. This is not gonna go through. This doesn't make sense. This individual doesn't exist. Okay, so this is how we condition on age, sex. So these are anthropometric data like age, sex, race, height, weight. Okay, and then what we do is that this could help us to get a mean individual, right? The mean individual given these conditions that we stated. Now, if we have an exact target individual, we can scale using something like this. So this is organ weight, the target organ weight, with just simple allometric scaling, okay? We have the mean height, we have the organ weight of the mean, and then we have an allometric scale, some exponents, so different exponents for different organs, and then we get the organ weight for the target individual, Okay, similarly, we do this with the cardiac output as well. So this is the simple idea, right? It's pretty straightforward. What happens in this larger loop is that this process is done over and over again. So we specify a range of these body weights or heights, and then we run over and over again, this create the individual loop until we get the required number of individuals in the population. Okay? There's also a bit of stochasticity added here. Like once you reach the target individual, you add a little bit of noise, a little bit of variability, because we also know like the coefficient of variation for these physiologies, because you don't expect two individuals with the same age and sex and height, for example, to have the same exact volumes or the same exact blood flows, right? So this adds this a little bit of stochasticity into the thing. All right, so simple, right? This is another algorithm, Huizinga algorithm, that does something similar, but rather than height, they base the scaling a little bit different, but they base the scaling on lean body weight. And their argument is that it's better than height, okay? And would you guys know why height, not weight, for example? Yeah, it's turned into a lecture, yeah. Someone's gonna raise their hand. So why not weight? Because at adulthood, height stabilizes, right? Together with most organs, okay? But weight could go up and down, right? So weight at adulthood might not be correlated with the organ volumes and the blood flows. Maybe the blood flows, so there might be an argument for blood flows, right? All right, so how do we do that? Well, rather than you guys having to dig through these literature references, sources, and putting these algorithms together, I did it for you. So I have this package, an R package called PBPK Toolkit that I put together and I shared publicly with the world. So this package would do that, right? So how do we generate, for example, a virtual individual? So it's on GitHub. It uses enhanced database for anthropometric data, which includes about 80K individuals. So it's a huge number of individuals, ages 2 to 80. So you notice here right away that it doesn't go to children below 2. So this is something that still needs to be appended to the algorithm. So it has the sexes, weights, heights, and BMIs. And if you run this, for example, it's very simple. It's an R function. And if you run this gen IND or generate individual, you give it the information like age, male or female, body weight, the target body weight, the height target, and then you optimize. So what's happening in there is that it's taking this information, looking at the slice of data through enhance. If the body weight or height are beyond the observed weights and heights for the chosen age and sex, it's gonna error. And it's gonna be a very informative error, telling you where the issue is. This optimize step, I chose to make it like optional here. What it does is that, because the algorithm, how the algorithm works is that it scales all the organs and then calculates the adipose as the difference between the target body weight and all the organ weights, okay? Which might not be very realistic. For example, if you have a heavy individual, you might have all the organs leaning to the smaller size, so you have a huge adipose tissue that is not realistic, right? So this optimization is does an additional optimization step to make the distributions of these volumes more realistic, which takes a little bit more time because it's optimization. So you see here that we generate all of these, and what the algorithm would give you is a named list of volumes and flows. And this is the representation here, as you see, all of these put together. And then the flow rates, okay? So, yeah, so something of interest to a lot of the audience here is that how can we generate the pediatric physiology, which is very simple. Use the same function, but put the pediatric anthropometric data, right? Again, it doesn't go below 2 years. And this is only going to scale things on the physiology, like the volumes and the flows, right? If there's any other consideration, you have to put it explicitly in your model. Like, for example, expression data, right? Enzyme expression data or what have you. Okay, if you look at generate the virtual population, this is the name of the function, genpop, and it does use that genint function under the hood, so it keeps calling that genint function until you have the correct number of individuals. So, for example, here we're generating 40 individuals. We're giving the ranges for ages, for body weights, for heights, right? Again, it's going to look at the enhanced data. If something falls beyond this range, it's going to discard, okay? And you can see that this is the correlation plot for the generated physiological parameters. And this makes sense, right? So the red means correlated. So you can see that all of the volumes are correlated, which is what we're looking for, okay? What about these ones? So the adipose seems to have like an inverse relationship to the rest of the organs. And this is just because of how we calculate it. It's the body weight minus the summed weights of all the other organs. So if the weights of the other organs are higher, adipose is going to be lower, okay? And also thymus, right? This makes sense. Thymus shrinks as we grow older. So that's reassuring that what we're getting is physiological. And then nothing very special about the blood flows. They are correlated. So beyond the volumes and flows, this is what we focused on so far. Beyond volumes and flows, there are other things that you might want to consider as well, like enzyme expression, like CYPs and UGTs, metabolizing enzymes, transporters in different organs, binding and tissue composition as well, like plasma proteins, tissue composition that impacts the partitioning of the drug, right? So these are additional things that you need to consider as well. So, yeah, been trying to put these also into the algorithm, but never had enough time. So if you're available, let me know if you want to collaborate. All right, so other things that the package can do is actually predict partition coefficients. So as we said, partition coefficients, these are like drug specific. They are the partitioning between the tissue and plasma, okay? And how are you going to get these partition coefficients to build a PBPK model? Well, you can grab a bunch of people, give them all drugs possible, kill them all, harvest their organs, and measure how much drug is there, right? This is, of course, very dramatic, but you know, it's impractical, but there are other imaging techniques that you can use also to monitor the concentrations of drug in different organs. Other things that you can do is do like in vivo studies on animals and just use that as an approximation, right? However, in practice, what people would mostly do is that they use in silico prediction methods. These are prediction methods that are based on things that are easy to measure in the lab for the drug, like pKa or lipophilicity of the drug. So these are much more readily available, and you can use them in these algorithms to predict partition coefficients, okay? So do you need to go through the different partition coefficients algorithms and put them all in R? No, because I did it for you guys, just for you. So in the PBPK Toolkit, we have the different methods that we actually published in this paper, the quantification of the impact of partition coefficients prediction methods on physiologically based pharmacokinetic model output using a standardized tissue composition. Also, another thing that we noticed in like software tools available to do this is that when you change the method, you cannot really directly compare the different methods, at least for the tools that I've seen, because when you switch the method, it switches the tissue composition as well, based on what tissue composition was published with that method. And by tissue composition, I mean the fraction of water, the fraction of lipids, and stuff like that in a specific organ, okay? So, yeah, we didn't like that very much. So we came up with our own standardized tissue composition that we compared with all the different partition coefficient methods, and it works well, and the predictions agree well with the ones that were published in the respective publications. And the outcome from this study is that there is no one method that is better than others, right? You can just put it as part of your optimization of your workflow. Just try different methods and see what makes more sense in your prediction, how it's impacting your prediction. And yeah, don't mind this, you're not gonna see this, but this is the tissue composition. That was not the purpose, Stacy, that's not the purpose. So this is upset because I have something here that is not readable, which, yeah, I agree. But this is the tissue composition, you can see it in the publication. All right, yeah, and this is the function that you can use. The function, it takes these things like log P, which is a measurement of lipophilicity, pKa, the unbound fraction in plasma, blood to plasma concentration ratio, type, which is like the type of the molecule, whether it's neutral, monoprotic acid, monoprotic base, dibase, stuff like that. I never liked organic chemistry. Method, Pauline and Thale, or PT, Pauline and Thale, or the other Huizinga for the Huizinga method, okay? Now the one I tested the most was the Pauline, these are the partition coefficients. These are not the generate individual methods or the physiology. So thank you guys for correcting me on that. So Pauline and Thale or 4 other methods are available in this function. Miscellaneous functions. So other functions that are also very useful, like calculating the blood to plasma concentration ratio. Okay, fun story, the first PBPK model I worked on was on voriconazole, and I'm still looking for the BP for that drug to this day. So if you have it, let me know. So something like this would have been very useful at the time. Also calculate the unbound fraction. So these are not like guaranteed to be that accurate, but it at least gives you something to start with. All right, so moving on from small molecules to larger molecules like monoclonal antibodies. Can we apply the same principles on monoclonal antibodies? How are we doing on time? We're good? Okay, faster. So, faster. Monoclonal antibodies, okay? People are much more interested these days in larger molecules, right? With TCEs and ADCs and all of that. So, something or a PBPK model for monoclonal antibody like this one is, you can see it's much more complicated. Each organ has its own sub-compartments, and you can monitor like the monoclonal antibodies going through the different stages and what have you. However, the principles still hold because a lot of these sub-compartments are scaled based on the volume of the whole organ, right? So if you scale that, all of the other sub-compartments are going to scale similarly, okay? So I also have gen-ind mab and gen-pop mab that would do the same thing for monoclonal antibodies. And by the way, this works well. You can apply the output from these functions on any tool that you're interested in using, but these work well with MRGsolve if you're familiar with that tool. It's just a general purpose ODE simulator. And to just finish things up with this example that we used our functions before it became a package, this GenMab, for the GenMab functions, we use them actually with this publication where we were working with Eli Lilly on the development of the first monoclonal antibody. It was the first one that went into clinical trials in the world. And these were crazy times, right? So we already had the PBPK model. We had some nice mean predictions, but the guidance from the leadership from Lilly was that we want to get the dose for the drug. We helped them with the candidate selection and the dose selection. We want to get the dose that would treat most patients, right? So to do that, we needed to introduce the virtual population, to generate the population and to see whether we cover like 90% of the individuals, okay? So that's what we did here, and it worked quite well. And yeah, actually, when our predictions, so the gray shaded region here is the predictions with the virtual populations. Predictions. This was before we had any like animal data or we had any human data. But then when Lilly people did the clinical trials and overlaid the predictions on the human predictions, on the human observations, it was just spot on. So it was like it was fit, but this was no fitting. It was just extrapolation. All right, so takeaway. I don't have a slide for takeaway, but I'm just gonna say it. Please use the tool and tell me if you love it or hate it. If you're willing to collaborate, let me know. This is my contact. So thank you.
Thank you, Ahmed. We have one online question really quickly.
Sure.
Is there a way to modify your parameters to choose, say, select a specific disease state population?
Yeah, it's gonna be like after the fact. So yeah, if it impacts like flows or volumes, yeah. If it's something else, then this will have to be in the model itself, like metabolism, if there's a condition in the liver, for example. You're welcome.
So I understand the functions that you presented for generating those virtual populations for those distributions of body organs, flows, and others are based on this 80K populations of observed measurements from actual individuals, right?
Yeah.
So when you use that function, is it something that is built in, or you can basically translate your algorithm to other software that does not have access to that data?
Yeah, we actually had like discussions. We never went through the whole like application, but we had like discussions on how we can implement some of this on the actual, on some, a tool that is out there. But yeah, I don't see why. It will work quite well if you have like a general ODE simulator or a general purpose ODE simulator. That's very straightforward. For other like software tools, it's gonna take a little bit more work.
Yeah, but you still need a database, but then if the data increases over time, let's say 10 years from now, you will have more information, you can add it, and you see that's what creates some updates, issues, this kind of thing. But when you just separate it, it will be just once forever, and then that's a value, at least for me.
Yeah, and this actually, because these databases are already available as R packages, so there is the code. Like I do have the code, and I think I put it somewhere in the package as well, the code that would generate these based on the database. So if there's an update in the database, you can just have another, locked down version with this update. Right? All right, thank you.